Hi, this is Roger Green, host of the Surfing the NASH Tsunami podcast. This weekend, we're offering 5 conversations from episode 60, our special coverage of Madrigal Pharmaceuticals' release of MAESTRO-NASH Phase 3 data, originally posted this past Wednesday. This conversation continues with the larger group and starts with Jörn Schattenberg asking the impact that the use of a broad set of NITs in this study will have on NIT adoption and its use in clinical patient treatment, not just clinical trials. Mazen Noureddin points out how much use of NITs in clinical trials can reduce recruitment time and costs associated with those trials. Scott Friedman points out that as we strive to learn how to identify drug failures better and faster, FDA might require broad NIT use in clinical trials as part of that effort. At this point, Rachel Zayas and Scott Friedman jump in to address some more basic scientific questions. Rachel asks whether there's a synergistic effect between LDL lowering and the primary endpoints in these trials, and if so, what that would mean for the disease. Well, or if not, what that would mean for the disease as well. Scott asks what these results tell us on 2 fronts. First, what does it tell us about the ability of fat lowering to regress fibrosis if THR-beta has no direct anti-fibrotic effect, but proves regression of fibrosis comparable to its effect on steatosis? Second, what about this drug converts fat lowering into fibrosis regression when other drugs that can lower fat and maintain lower fat levels do not demonstrate such rapid anti-fibrotic effects? After more comments about the science of the trial, Mike Patel asks how quickly focus will shift from monotherapy to combination therapy, given how many patients did not succeed with Resmetirom as monotherapy. Mazen notes that other therapies were administered to patients in this trial, and as the conversation ends, Louise Campbell asks about semaglutide design and how it relates to this trial, and Mazen discusses both benefits and further trial opportunities for semaglutide. Here in Nashville, where we all live, the world changed for good on Monday. This is a quick take on some of what that change might look like. It will be interesting in years ahead to look back on this conversation to see what we got right and wrong, and you get a sneak peek right now. So sit back, listen, enjoy, learn, and when you're done, join the conversation in our LinkedIn discussion group.
Jörn Schattenberg.
What does this trial mean for NITs? Because it's particularly rich in non-invasive tests, and we're seeing response. And the question came up, who does respond? So, the take on, we're going to see a lot of compendium diagnostics, I think, here, which might be qualified, hopefully.
Mazen Noureddin.
Yeah, but if you remember a lot of, the GLP-1 data, they do studies on multiple populations, as you said. Some of them, they don't have cardiovascular risk factors to start with, and they see if there are any effects. And some of them, they did have cardiovascular comorbidities, and if they see if there's any effect. So your point is well taken, actually. I do wonder if we'll see anything, but it's just encouraging to see such a data. And if there's a signal at the end of 4 or 5 years, I'll be very excited about it. And once we get through the liver biopsy, we can do 5 to 10,000 cohort data and look at MACE and non-MACE and the points that we wish.
Scott Friedman.
It may well be that the FDA at some point will require them because, as I mentioned, nobody wants the patient to remain on a drug that isn't working. Most of all the patients, but also the payers. So a lot to be learned still.
Rachel Zayas.
Well, this might be a good transition for Scott, because one thing I found of particular interest for me is in addition to the primary endpoints, the secondary endpoint with the lowering of LDL cholesterol. And it really begs the question, if the patients who had hit the primary endpoint, what % of them specifically had the secondary endpoint as well? And it begs the question, is this a synergistic effect? And what other secondary endpoints should we be monitoring and assessing? So I know that this was top-line data, but I'm really interested to see what comes out of this from that perspective.
So in addition to all the exciting implications we've talked about on the clinical side, for me it's very interesting in terms of the mechanism of action of this drug, because one of the unanswered questions is lowering fat alone good enough, number one, and I mean good enough to reduce fibrosis and get hard endpoints, and number two, does it matter how you lower fat? And the answer to number one, of course, still is not completely known, but it moves us towards a conclusion that reducing fat may be a good thing. I say that because, to my knowledge, the drug has no direct antifibrotic effect. And so any benefit in fibrosis and NASH resolution was largely, if not exclusively, based on its metabolic impact on hepatocytes and the reduction in the signals that drive inflammation and fibrosis. So I think clearly more studies to be done. With regard to the second question, I actually still would not assume that that any way you lower fat is the same. Because there are drugs that lower fat that probably are not going to be as effective, even though the net result of them is that the fat lowering is consistent. So it may well be that lowering fat is necessary but not sufficient. You have to lower fat and create the right metabolic milieu or reduce the production of molecules that drive inflammation and fibrosis, and that may not be true from every mechanism in which fat is reduced. So again, lots of scientific questions here to tackle as well.
And the critics to the approve as just to the approach, as Scott just said, was always said, it's not enough to improve lipids. Statins do that, no effect, and here we are today seeing NASH resolution and fibrosis regression. So I echo Scott's comment. We want to drill down on the link of, and I think there are a lot of links in the literature, lipotoxicity, activation of stellate cells, and all the work Scott's been doing over the years. So it seems to read out in a rough clinical trial. So that is fascinating.
And also, along the way, when their phase 2 study came out, they did not meet fibrosis improvement, and obviously the study was not powered to do so. But along the road, we started seeing NITs from their other MAESTRO-NAFLD data and others showing that the stiffness measurements are improving. MRE was one of them, MAST was one of them, transient elastography was one of them. So it's really good here to see that the NIT started predicting all along that fibrosis improvement prior to have adequately powered study. Just adding to the evidence that NITs biologically make a lot of sense because they represent a physiological process.
Yeah, speaking of drilling down, one other implication of this is to go back, and we all agree we should move past biopsy, but for as long as we have biopsy, let's start applying or expanding our efforts to apply digital pathology and artificial intelligence to see if there are patterns or subtle changes that were unmistakably present in the responders early on, or that there are other features that both give us a hint into new biology, but also define elements of the biopsy that may be more subtle than what pathologists can identify in further classifying or refining our assessment of the disease histologically. I mean, there's no question in my mind that any of the digital methods that are currently being developed are far more sensitive and informative than what a pathologist can tell us.
So 2 things on that. First of all, spoiler alert, Stephen, when talking about what's going to be presented at NASH Tag, talked about at least one presentation that will demonstrate for one drug the ability to use real simple liquid CBC level metrics to tell you relatively quickly whether that one drug is working or not. So I totally agree with Scott. We need to be able to figure out faster what doesn't work. Some of that might be very complex. Some of it might be simpler than we'd ever imagine. But yeah, it's something we need to get after immediately. And speaking of simple, I thought the most interesting thing about selecting LDL out of all the secondary endpoints to be the one to focus on today had nothing to do with anything we're talking about right now, but had to do with messaging the investment community that if the problems with OCA were pruritus and LDL, there's no pruritus here, and LDL, in fact, was a positive, not a negative. So going back not to the science of it, but to all the directions in which this message is out, I thought that was a really interesting decision. And by the way, the fact that we are now spending so much time talking about LDL makes that decision look really good because it integrates it into how people think about the profile of the drug. And I think that's a big step forward for how we broadly conceptualize what we can expect to see with drugs in this class. It's really interesting, all the different levels on which this stuff operates.
Yeah, the comparison with HCC is interesting, but I think as Mazen mentioned before, we have to remind that this is not going to be standard of care until we have full approval. So that's, I think, in the way you design your trials. Of course, you want to think about, if you start now, when are you going to be reporting out, and what's that going to mean? But for all the ongoing trials, and also the second thing, as Scott mentioned rightfully, you got to show efficacy for each drug explored in combination in monotherapy versus placebo. So there will be more monotherapy, I think.
Mike Bottel.
I have a question for the group. Just wondering, as we look forward now, so now there's a new bar, And we talked about that 26% wasn't really there yet. So do you think that combo therapy is going to be the next one before another monotherapy comes out that's better?
I think the example we can look to is actually from the liver cancer or the cancer community, where a drug is approved, then it's compared for non-inferiority to a second drug. At the same time, the next generation is take the first drug that's effective and add on a new drug with a divergent or complementary mechanism. And I would imagine we're not done with single drug therapies at all, but it's almost inevitable that given the response rate of 24% that we're going to be rapidly, and I'm sure more than anyone, Madrigal will be very interested in amping up the effectiveness of their drug by adding a second agent. I'm sure there'll be a lot of recruiters who are auditioning for that role.
Yeah, that does not preclude the company, as Scott suggested, from looking for a combination for added therapy, because they're expected to be approved and they just want to build on that momentum and keep going. He's right. I think there will be multiple drugs tested to see if they're efficacious before they started to be added on to Resmetirom. But if I'm Resmetirom or Madrigal, I will probably now look into combining with a GLP-1, combining with SGLT2s. I also want to remind the listeners that there were actually people on GLP-1 in this study. There were, yes, about 14 to 15%, and so as SGL2. So the Resmetirom seems to add to that in such a group, or probably not. We need to see the analysis, but still used, yes. But it will, I think even the sub-analysis will be interesting, but probably not enough numbers. But eventually, I would love to see it with systemic drugs or systemic drugs with an effect on the liver, such as the dual GLP glucagons, and adding Resmetirom, because you want to think about it as the first approved or one of the two first approved therapies for NASH.
Louise Campbell.
Can I ask, and I know hindsight is a beautiful thing, Novo Nordisk and semaglutide, obviously approved in diabetes and obesity medicine. Did they miss a trick by actually not really using non-invasive technologies for their liver health, given the high-risk populations that they used, and liraglutide, where they could have been collecting data for these studies? Or is it something that they could do protocol deviations for now, or protocol amendments, not deviations, to put into a later phase study within the populations that they've got? Because obviously it's going through the NASH, but obviously there's NAFLD populations that they're treating who would have had NASH. And the recent semaglutide patients that I've seen have all got perfectly healthy livers on FibroScan, and I know that's only one assessment.
I think they have a transient elastography in their study, not in the entire population. At the end of the day, they are getting NASH patients with biopsy-proven fibrosis. So I would say I would love to have more NITs, not just because I'm an IT person and I like NITs. I would like eventually to see a treatment response and pushing the field forward. But semaglutide has incredible efficacy as well as systemic effects and incredible advantage. To answer your question, maybe having more NITs is better, but are there disadvantages? I don't think so. It's a very efficacious drug. I would love, as a doctor and some patient advocate, I would love to see semaglutide with Resmetirom combined. They will be, before people starting doing big phase 2s and phase 3s, there are investigator-initiated studies and smaller studies proof of concept. So hopefully the small parties of combo drugs or small study, investigator-initiated study, few centers will start occurring in this space.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please send an email to questions@surfingnash.com. We will be back next week with the rest of our year-end reviews. 2 episodes that include conversations with Mazen Noureddin, Ken Kuzi, Jeff Lazarus, Stephen Harrison, Naeem Al-Khouri, and Ian Rowe. You'll want to hear all of it. Until then, wishing all of you a wonderful holiday season and best wishes for a fantastic 2023. Stay safe, surf on. We'll see you next year on the podcast. Bye-bye now.